Okay. Okay. So, hi guys, this is the Biology 285 review session for week 3, and we're gonna get started. So one of the first things we spoke about was where proteins are made. Hi, Melina. Hello. Melina, the other TA. So for eukaryotic cells, proteins are made in the cytoplasm by ribosomes. And there are two types of ribosomes that we discussed in class. Sorry, I'm blocking this. Two types of ribosomes we discussed in class. You have your freely floating ribosomes that just float around the cytosol. And then another kind we have is ribosomes that are attached to the membrane of the ER, of the rough ER. And proteins that go into the endomembrane system are made by ribosomes that latch onto the ER membrane of the rough ER during translation. And I'm going to talk about the endomembrane system in the next few slides, but these are just examples of things that are part of the endomembrane system, things that have a membrane, so the ER, lysosome, Golgi bodies, etc. And proteins that don't end up going through the endomembrane system, are made by ribosomes floating freely in the cytosol. So these are proteins that just stay in the cytosol, nuclear proteins, mitochondria, chloroplasts. Does anyone have any questions? I knew that was going to happen. So this is just a diagram of what I just said. You have the two types of ribosomes. You have your freely floating ribosomes and the membrane-bound ribosomes. And yeah, so as you can see on the right, you have proteins that end up going through the endomembrane system. They always start traveling through the ER. And so you see that here, and then the different destinations they can go to, depending on the signal sequence, which I'll get to. So just a visual representation of what I would say. Good? Give me a thumbs up, anyone. Thank you. So now I'm going to talk about the endomembrane system. An endomembrane system is essentially membrane-bound organelles that are able to participate in protein trafficking. Like I mentioned before, they have membrane-bound compartments such as the ER, Golgi, lysosome, cell membrane, and they have these special transport vesicles that move between them. And this is also referred to as the secretory pathway, just another way of saying the same thing. And any protein at all that functions and lives, quote unquote, in the ER, Golgi, lysosome, or being secreted out of the cell has to travel through the endomembrane system. So proteins that are going to those destinations have to travel this way. And that's all due to signal sequences. And just like I said before, all of the endomembrane system, all of the transportation of proteins that go through the endomembrane system has to begin in the ER. And yeah, I actually have a visual representation of this in the next slide. But essentially, the protein will go through the ER, and then it'll travel on these special vesicles and move towards the Golgi, and then it goes through the Golgi and tapers off into a vesicle that attaches to the cell membrane if it's going to go outside of the cell. And this is something I think Laura mentioned in one of her videos, but if a protein only has an ER localization sequence, it may be able to continue to travel until it gets secreted out of the cell. And it won't stop unless it has some other sequence telling it where to go. So if a protein just had an ER localization sequence, it would begin its journey through the endomembrane system. And if you wanted your protein to stay in the ER, it would have to have the ER import localization sequence as well as an ER retention sequence. So it'd have to have two sequences to tell the protein that, hey, you're supposed to work and function in the ER, stop here. So if it only had ER localization and no retention sequences, it could continue floating through the endomembrane system until it reaches the end, which is being secreted out of the cell membrane. Does anyone have any questions on anything I said? No. Okay. I'm sorry, I can't see. Yeah.
So this is like kind of like a question on concept.
Okay.
It's asking,
No, go ahead. Sorry.
it says there's like ER signal sequence, but also, and it's read first because it's that on the side, but then there's also like an L. So is that just not?
So I'm actually gonna, I think like it'll be answered in the next few slides. But yeah, I'm gonna go to the next few slides, see if that helps. But if it doesn't help, then remind me at the end. We can go through that question. So yeah, again, it's just another visual representation because I like to look at pictures as opposed to reading words sometimes. But essentially, the endomembrane system is kind of upside down. So you have the ribosomes that are docked to the rough ER that proteins that are synthesized go through. And then there are these special transport vesicles that move proteins from the ER to the Golgi. Then the proteins go through the Golgi and they taper off into these vesicles and go towards the cell membrane. The vesicle is able to fuse to the cell membrane and the protein can get exported out of the cell. This is just something I added in here just to reiterate what I said before. But anytime a protein is localized to the ER, it has to have an ER signal sequence. And if it doesn't have a sequence, it can't enter. And this is because the ER signal sequence is recognized by SRP, which is a special particle that will attach to the sequence, and the sequence is N-terminus, like you said. I am going to get to more of the details on this in the next few slides, but the signal sequence is on the N-terminus, so towards the beginning of the polypeptide, and that what am I looking for? That sequence of amino acids or that special chain is read by that particle, so it's able to bind there and bring the protein into the ER. And so that's why like not having that sequence, if it's missing, then the protein will never be able to get trafficked to the ER. And like I also mentioned, if a protein has to go outside the cell, it has to pass through the endomembrane system. And once a protein leaves the ER, they rarely come back in again. I think this was discussed in the UPR response a little bit. And the process in which proteins return to the ER is called ERAD, and it's for degradation. But I don't think she went into much detail about that last point. Now I'm going to talk about how do proteins know where to go, and I mentioned it earlier, has to do with signal sequences. And signal sequences are usually contained in the amino acid sequence of a protein. I try to make various analogies in this class. So I would say, think of a signal sequence as a unique zip code. So like a zip code tells you where a destination is. So when you're shipping a package, you put the zip code for Amherst 01003. And so you know that like that package is going to go to Amherst. And signal sequences are not added, is not added after a protein is being made. This is very important because it's what distinguishes it from post-translational modifications. So signal sequences are built into the protein. That's why I said it's embedded in the amino acid sequence. And so, yeah, I think I mentioned that point 3 times now. But yeah, so when a protein is localized to the ER, has to have the ER signal sequence. And this is, again, just because of the SRP, which can bind to that protein and ship it off to the ER. And if it's missing the ER signal sequence, then it doesn't have that binding site that's recognized by SRP. So therefore, it can't leave the cytosol. So it will stay in the cytosol after it's been translated. And yeah, I'm sorry, I think I just repeated myself there, but I mentioned that before. And I'm gonna actually post these slides at the end of the review. So if I flip through them quickly, don't panic, I'm gonna post them. This is a chart that I got from my 151 class. I don't know if any of you had Professor Phyllis, but this is where that chart is from. But it's just like a visual way. Sure, you can take a picture, but I'm gonna give you the slides. This is just a visual way to, I always send the slides, but this is just a visual way to look at signal sequences and a way for you to practice as well. So if it had an ER signal sequence, it's gonna go to the ER. And then if it, I would ignore the anchor sequence. I don't know if she talks much about that, but anchor sequence has to do with the protein being like sort of like a transmembrane protein. But so if a protein needed to get out of the cell, you kind of work through the sequence. So does it have an ER localization sequence? Yes, because it has to pass through the endomembrane system. And then it doesn't have an anchor sequence. So it continued through the ER lumen, then through the Golgi bodies. And then as you can see, if it doesn't have any other signal sequence other than the ER signal sequence, the protein ends up being secreted outside of the cell. So does that make sense? Okay, and then if it didn't have an ER signal sequence, then the protein is made on a freely floating ribosome, and so it can end up in these other places, which I will get to in the next couple of slides. Okay. More about signal sequences. So signal sequences act as a binding site for another protein to bind to. An example of this is when you look at the nuclear protein transport, proteins that end up working in the nucleus. How do they get there? It's through this special protein called important that binds to the NLS, which is nuclear localization sequence. And that sequence is on the exterior surface of a protein. And so important can recognize that as depicted here and bind to that binding site or the signal sequence and bring it through the nuclear pore. Interesting thing to note about nuclear proteins is that they can enter the nucleus fully constructed. So they're not unraveled like spaghetti, as you see in some of the other diagrams for ER transport and mitochondria transport. So that's like a difference between the two. So when I say that a protein enters the organelle fully folded and fully made, it means that it enters in its tertiary structure. So it's 3D shape. And the reason why nuclear proteins can do this is because the nucleus has special nuclear pores that are able to accommodate for the size of these proteins. And so the protein is just able to kind of wiggle through these pores here. Yeah, and so a difference between the way proteins go to the nucleus and proteins go to the ER is that the ER and mitochondria have these N-terminal sequences. If you guys remember from week 1, like the N-terminus is towards the beginning of a polypeptide and C-terminus is towards the end. So N-terminal sequences are on ER and mitochondria proteins, and they usually end up being cut off. So once the protein is brought into that organelle, so the ER and mitochondria, so it doesn't exit the organelle. But I'm going to go through more details on that in the next slide. Does anyone have any questions? Email. Yeah, I always try to make sure, or whichever TA is doing the reviews, that we email them. Yeah, so yeah, I'll make sure they'll be emailed. So don't panic if you didn't write stuff down or notes.
So the signal sequence for nuclear is it clean? Is that because it can't be because it's like embedded in the protein already, or because like the protein needs to be able to come out of the sequence?
I'm not sure the reason why it doesn't get cleaved, but the difference with the nuclear proteins is that that signal sequence is on the exterior surface of the protein. So in its tertiary structure, it's just kind of like on the surface. So that would be kind of difficult to cleave. My guess is, Lena, do you
know if it matters, like if it's important to be there because the proteins have to like be able to
so I know for the nuclear proteins that they have to have a different signal sequence to leave the nucleus. So I think maybe I didn't clarify before, but the nuclear localization sequence is to take a protein from the cytosol to the nucleus. But then there's also a different signal sequence that brings the protein from the nucleus back out to the cytosol.
Is
And
that
then,
the same, sorry, is the same signal sequence for like, cell division?
I'm
Or is
not
there
sure.
no sequence?
For cell division, there's different sequences that will initiate that. For cell division, that goes more into gene expression, which I will cover more on later in the slides coming up after Michelle's done. Sorry. But what I was gonna say is that like with proteins that are supposed to go in and out of the nucleus, they're already like fully made proteins. So like nothing is going to be cleaved on or off of proteins or anything like that. The only time like cleaving kind of happens, I believe, is during RNA processing. And that's when like parts of the like the exons will be left in, but the introns will be cleaved out. So that's the only time things will be like kind of cleaved on and off. But you're right that the nuclear localization sequence, that's a sequence that's on the protein, that will never be cleaved off. And that is definitely something that's necessary for the protein to be moved into the nucleus. And Michelle is right that there is another sequence called the nuclear NES,
nuclear
export sequence. And that sequence is necessary to be in the protein sequence in order for that protein to leave the nucleus. But not all proteins have both of them.
Sorry, you guys will hear from Melina. I just split the slide, so like I'm doing like the first section and she's gonna do the other like half, but you will hear from her. I know it's a lot of me right now, but I wanted to mention that when we go over, did you guys talk about nuclear import and export, the cycle? Is that part of this week's? Yeah, I believe so. Okay, so we're gonna go through, once you guys learn about signal sequences, it's gonna make sense because there are certain proteins in that cycle that exit the nucleus and go to the cytosol and go in. So something like exportin would have to have a nuclear export sequence to be going from the nucleus to the cytosol, and then an NLS, I think, as well. But then important, because it lives in the nucleus, just has to have NLS, which might make a little bit more sense once we get there. But did that make sense for now, for what we were talking about with your question?
Yes.
Okay. Yeah, and Melina, feel free to add anything. If there's anything that I'm like saying wrong, or whatever, or if you have any extra stuff. So just more diagrams of protein localization. I just like to put diagrams there for people who have different learning styles. And I know there's a lot of things, sorry, 1 second. I know there's a lot of things to remember about the different types of localization areas that the proteins go to. So I made a little summary chart on like the differences and similarities between certain kinds. And so for the nucleus, you have signal sequence that has to be on the protein at all times. Compare that to the ER mitochondria, signal sequences gets clipped by a special protein called signal peptidase. Not sure if you have to memorize that name of the protein, but just being aware the signal sequence gets clipped off after the protein enters the organelle, and that's to prevent the protein from flying back out again. And as you can see with the ER and mitochondria, your signal sequences are located in the N terminus of the polypeptide. So at the beginning of that protein. So once the protein enters the organelle, it will kind of, the polypeptide chain will kind of look like spaghetti. So that's what I mean when I say enters the organelle unfolded. So enter while the protein's being like synthesized through the ER. And that's because when a protein is being synthesized, the N terminus is created first. So as soon as, sorry, I thought it was my phone. So when a protein is being synthesized, the N terminus is made first. And as soon as the N terminus is created by the ribosome, the SRP signal is quickly recognizing the signal sequence in the N terminus. So it's already trying to pull that protein into the ER while the rest of it's being synthesized. So in the diagrams, where's my pictures? In the diagrams, you see it being pulled in like spaghetti, but that's because that N-terminus sequence is being recognized by SRP and it's being brought into the ER. And then once it's inside the ER, chaperone proteins help to refold that protein into its correct 3D structure. And that signal sequence is cleaved so that it's not able to leave. Sorry, it's flipping too fast. So yeah, so those are common differences between nucleus, ER, and mitochondria. And another thing I was going to mention, which I think I did, was the nucleus. Remember that the proteins enter fully folded and fully made, so in their proper 3D orientation, whereas ER and mitochondria proteins enter unfolded, so like strung like spaghetti, and you have chaperone proteins inside that are ready to refold it in the correct shape. And for a nucleus, the signal sequence is on the outer surface, the exterior of the protein, whereas for ER and mitochondria, the signal sequence is located in the N-terminus. So it's just towards the beginning of the protein. And as soon as that protein is being made, it's already being pulled into simultaneously at the same time as it's being synthesized, the N-terminus is being dragged into the ER and mitochondria. So those sequences of amino acids is recognized instantly by SRP. And that's what I mean when I say that the signal sequence is embedded in the primary structure. So those special amino acids correspond with the signal sequence. Did that make sense for people? Can I get head nods or thumbs up? Thank you. Any questions? No? Okay. And more diagrams, because why not? So basically what I was saying, you have SRP recognizing, looks like this pink structure here is the signal sequence. And as soon as that protein is being made, as soon as that N-terminus comes out, SRP binds to it. And that protein hasn't finished being made because the ribosome is still trying to make it. So it's being attached, it's being pulled right into the ER. So, sorry, I was pointing at the wrong thing. It's here. So while that protein is still being created, it's already making its way into the ER. And then once it's inside the ER, the protein cleaves that signal sequence off and is able to stay in the ER. And because the protein enters unfolded, it gets refolded by chaperone proteins. And yeah, so that's just a diagram of that. And the mitochondria, which sort of happens in a similar way. Okay, so there are different types of signal sequences for this class. You don't really have to memorize the specific amino acids that are involved in them, but just having a general idea about these things is a good idea. So for ER import, you have like hydrophobic amino acids, generally speaking, that are part of the signal sequences. And it's important that the signal sequence is created this way so that SRP can recognize it, because SRP recognizes a specific type of hydrophobic amino acids. And if you could imagine that if you were asked a question where you tweaked the amino acids so that you put in hydrophilic or polar amino acids, you may be able to draw some conclusions about what could possibly happen or not happen with ER import, hint, hint. Another thing to know about different types of signal sequences, which I brought up before, but for a protein to function in the ER, it has to have ER import because that gets it into the ER, and it has to have ER retention sequence. So if that's a protein's final destination, so if a protein is going to work and its destination is the ER, it has to have the signal that tells it to enter the ER. So that's your ER import localization sequence. But then it has to have the sequence that says stop here, you're staying here, this is your destination. So that's the ER retention sequence. And the ER retention sequence is usually located in the C-terminus. So that's what I mean when I say protein is going to live and work in the ER lumen, meaning that it's going to stay there and be ER protein. It has to have both. So she might try to trick you and ask you if a protein has just the import to the ER sequence, it will stay in the ER. And you can be like, no, it has to have both the ER import and the ER retention sequence. So that's how that works. Mitochondria, typically there's interspersed positively charged amino acids. She generally doesn't like ask you to memorize like the type of chemistry, but just I'd say having a good idea of the ER import tends to have hydrophobic amino acids, so disturbing that could have some effects. And remembering that ER proteins have to have 2 signal sequences for them to be able to function in the ER. I think those are very important. But yeah, I don't think she asked, don't worry about knowing exactly what kind of amino acid, like alanine, valine, whatever that is in the signal sequence, like that chart she showed you in class, which I have on the next slide. You don't have to memorize the specific amino acids, but having the idea of the chemistry, especially for the ER ones, I'd say. And yeah, it's for signal sequences for the nucleus, it doesn't get cleaved, and it's important because it's a binding site for important, so it has to bind there. And the signal sequence appears on the exterior surface of the protein. So if you were to manipulate a protein or change that sequence, you could predict what could or could not happen. And same idea with the export NES sequence. And for the cytosol, I thought I would list it there because I think she went over this in class. Proteins are created in the cytosol. So therefore, the protein is made in the cytosol. It's made by ribosomes floating in the cytosol. So it doesn't need a signal sequence to get there because that's where it starts. That's where it begins its life. So there is no signal sequence for cytosolic proteins. And any proteins that have 0 signal sequences, they are here where they are made. So does that make sense? So this is the chart I was talking about. So do not memorize any of these amino acids because you don't have to. Okay. Yep.
So I just want to clarify, so importing, exporting are only.
That's right. Important and exporting are only for nuclear proteins. Good question. Yep.
Could you classify like
Hmm?
as
I want to say SRP specific to the ER only. It's not, I don't think import and export are a type of SRP. And I don't think she goes through that in class. Yeah, so for this class, I would just know that SRP link that to ER proteins, and an important and exportant are their own group. Yeah, I don't think it's an example of that. But even if it was, you wouldn't need to for this class, know that. Okay, it's not. I don't want to confuse people who watch it on YouTube later. Okay, so I put a practice question in here. I rarely do practice questions, but I thought it would be a good idea. So we'll see how that goes. So I'm gonna give you guys like 2 minutes to read it. I just feel like you guys can read. I don't want to just, you're gonna hear my voice for the next couple minutes. So you guys can read that for 2 minutes and then we're gonna go through it. Do you want? If you want. You could take it over. Okay. Does anyone want to try to take a guess where they think this scientifically engineered protein would end up?
I think it would be cytoplasm or the cytosol,
Okay.
because if you're moving the signal sequence, the ER signal sequence, then it's not going to have any other signal sequences. And proteins that don't have any sequences will stay in the cytosol after they're made.
Yes. Did people hear? Yes, you got it. Yes. Okay. Sorry. You'd expect to find a protein in the cytosol. Yeah, you got it right. Okay. So it just based off of like the mechanism I was talking about. You have to have the ER, that should typo, should say ER import sequence, like the localization one, the one that brings it to the ER for the SRP to bind to it and bring it into the ER. And if you don't have that signal or any other signals, the protein is going to remain in the place it started its life. And that place is the cytoplasm. So you got it. It looked like a tricky question at first, right? But when you took a couple minutes to think about it, made sense. But yeah, that's an example of, I think that's a question on our problem set when I took the class. I took it 2 years ago. So just a cool example. And another analogy, because I actually love to use analogies all the time. So this one just is about a common life thing. Not sure with the pandemic how often people have done this now, but when you get a ride from Uber, if you don't tell the driver your location using your location services or GPS signaling, then they'll never find you and take you to your destination. So it's the same way with signal sequences. There's no ER import sequence on a newly made protein, then there's no way for SRP to recognize the protein and bring it to its final destination. So as you can see, like SRP is kind of just like the Uber for the ER protein. Did that make sense? I always try to put things in real life, and sometimes it works, sometimes it's silly, but
I like that.
you like it? Thank you. Yeah, and I hope you guys on YouTube watching it later like it too. Okay. Wait, what was this slide from? What was I trying to connect it to? Oh, so this protein in this question is example of a transmembrane protein. But we will get to those when we talk about pathways. But that's not none of your concern for now. This is probably like exam 2 stuff. Exam 2 stuff. So like week 6, I want to say. Yeah. So cool stuff. And oh, I forgot. I thought it was gonna say Melina's name on there. That's why I'm like, oh no, I have one more section and then I'll hand everything over to Melina. You guys have done this extensively. Okay, so secretory pathways. So Laura showed you guys in class an example of how protein trafficking happens in real life, but this example is just using hormones. Looks like it can be applied to insulin signaling, and it's pretty much the same idea, protein sorting and trafficking. I can't remember if hormones are considered proteins, some of them, or yeah, Melina's nodding her head. Yeah, so just a real life example. It's always nice to put things in real life. And another example is the lysosomal enzyme, which we talked about in class. And yeah, so just examples and pictures. Travels through the endomembrane system because lysozyme is one of those. I'm not sure if lysozyme is part of the, I don't know if it's part of the endomembrane system. And no, it is part of the endomembrane system, excuse me, but I'm trying to remember if it's like a membrane bound. Yeah, it is. Yeah, sorry. I don't know why I'm doubting myself now. But yes, it is part of the endomembrane system. Okay, so now the other half of the review will be taught by my good friend Melina.
Thank you, Fran. Hi, I'm Melina. I'm the other undergraduate TA. Hi, people on YouTube. So we're going to talk about nuclear import and export, and there's some really nice diagrams to explain it. But before I talk about nuclear import and export, there's this really important part of it that I'll go into, GTPases. So there is a GTPase called RAN. RAN is an enzyme, and all enzymes are also proteins, but not all proteins are enzymes. That makes sense. So RAN is a protein and an enzyme, and its job is to okay. So one of RAN's cousins or siblings is RAS, which there is a diagram of later on. But basically all you need to know is that the job of RAM when it's active is to cleave GTP, which it is bound to when it's active. But when it's inactive, it will be bound to DP. And I think that's basically all you need to know about this slide. RAS and RAN, they both have the same function to have an active form and then an inactive form. And its active form is when it's bound to GTP, and its inactive form is when it's bound to GDP. I think that's all for this slide.
Yeah, and sorry. You will learn about RAS when we do pathways coming up, but for now you'll see RAN, which is part of the nuclear import-export, and do not memorize the nuclear superfamily. I feel like I have to emphasize things do not memorize because I always get questions like that, but don't do it. Don't waste your time. Okay.
Mhm. Yes. So there are also proteins that help regulate what RAS and RAN are bound to, and they are called GEFs and GAPs, which are all proteins. And there is a helpful diagram after this one, that will help me explain this. But I'll just read through this first. So GEFs are guanine nucleotide exchange factors, and they help exchange GDP to GTP. Actually, let me just like explain this. I feel like this is easier to visualize. So you see how RAS is currently at the top, it's currently bound to GDP. That's its inactive form. GEF, which is another protein, the guanine exchange factor, it aids RAS to exchange GDP to GTP. So basically GEF helps switch that out, if that makes sense. So RAS, when it's bound to GTP with the help of GEF, it is now active. And so it can go on to do things within the nucleus, which I'll go into later. And then you have another protein called GAP. And GAP is a GTPase protein as well. And it aids in cleaving off one of the phosphates of GTP to transform it into GDP and make RAS inactive.
Sorry, I want to say one thing. You don't have to memorize what the like GEF, what it stands for, but we just put it in there so it can help you distinguish the differences between the two. Nucleotide exchange factor, exchanging one nucleotide for the other. And then GAP stands for GTPase activating protein, which can sound like it's doing its job activating, but if you think hard about it, GTPase is an enzyme, anything ending in ase, remember? So GTPase, the reaction that a GTPase does is hydrolyzing GTP into GDP. And you'll learn about this when we look at RAN in the nuclear import and export, but RAN itself is an enzyme and a protein. And because RAN is an enzyme, it has an intrinsic ability, meaning that it by itself can hydrolyze GTP into GDP, but it does so at a very slow rate. So GAP is a GTPase activating protein, which basically attaches to RAN or RAS, its cousin, and helps to accelerate this process. So it's not really GAP that's doing the hydrolyzing, because these enzymes like RAN have the ability to do it itself. So the wording of it, saying that GAP hydrolyzes GDP and a GDP would technically be wrong because of the fact that RAN can hydrolyze GTP and a GDP, but GAP just helps speed in this process up, which is useful for nuclear import and export, which has to happen over and over again. So just something to point out with the wording of it, because that is important. Yeah.
Can GEF, so I know RAN can, like you said, RAN can do the GAP's job on its own, just much slower. Can it do GEF's job on its own?
No.
Okay.
That's why GEF is there to help. Yeah.
Yeah, so a good thing to point out about GEF as well is that it helps with switching out GDP for GTP through binding. So GEF will bind to RAS in order to cause a conformational change in RAS or RAN. And that conformational change will cause GDP to no longer fit really well with RAS or RAN in their binding pocket. And so GDP will leave, and then GTP will go inside instead and bind because it has a better association.
sorry, I keep talking, but I just had another way to think about that. But so for GEF, what happens is when GEF binds to RAN, it can cause conformational shift in RAN that it loses its grip on GDP, or it has causes it to have a lower affinity, words from week 1 and week 2. So GEF can bind to RAN and cause it to have lower affinity for GDP. So GDP will no longer associate with the binding pocket of RAN. And so GDP will fall out of that binding pocket. And that leaves space for GTP to bind into that pocket. Because the binding of GEF to RAN causes a conformational change, which is essentially a shapeshift in the protein, which means it's no longer able to bind to GDP. So then GTP, since it's more abundant in the cell, will look for that empty binding pocket that has a new shape now and can fit GTP. And yeah, I'm sorry, another thing is that going from GDP into GDP to GTP, sorry, is just like tacking on another phosphate. Because remember, GDP is diphosphate and GTP is triphosphate. It's like adding on another phosphate.
So moving on to nuclear import. So I'll just talk through this diagram really quick. The words are basically just like what I'll be saying, but like kind of more clearly written. So if you start at step 1, which is all the way at the bottom, you have fully folded proteins and they have a sequence. On the diagram, it's called a cargo protein, and fully folded proteins with an NLS sequence, the nuclear localization sequence. Important is a protein that will recognize NLS, bind to the NLS sequence on nuclear proteins, and then bring them to the NPC. The NPC stands for nuclear core complex. And it's basically a complex of proteins that exists on the envelope of the nucleus in order to bring important and that cargo protein into the nucleus. So after it passes through the nuclear core complex, this is where RAN comes in. So RAN has to be active, and GEF aids in causing RAN to be active by binding to RAN and then changing a conformational shift in RAN so that GDP is switched out and instead GTP associates and binds with RAN instead. And so the RAN GTP complex then RAN that is active will now cause the protein and important to dissociate. And instead of RAN and GTP will bind to important and bring it outside of the cell. And so active RAN, which is bound to GTP and important, it will dissociate, it will actually cleave one of the phosphates off of GTP to become GTP. RAN will hydrolyze that GTP with the help of GAP. And so that is how RAN will then become inactive. Important will now dissociate from inactive RAN, and then important can go back and do its job. That was a little quick, but you guys are okay with that? Yes.
For the sake of all these problems, basically, is are RAN and RAS synonymous?
They're two. Sorry, go ahead.
I know one of them is in the super family of another one,
Yes.
but I see them being used like they're interchangeable.
For nuclear import, I would just focus that it's RAN, not RAS. RAS is in a different section. It's
We can discuss if it has to do with the problem set you said.
No, it's just in general.
In general.
Like, see RAN being mentioned and then RAS in similar ways.
Yeah, we were mentioning RAS earlier just because of the diagram.
It's because they're like both enzymes and protein, but they are two different things. So just when you think nuclear import and export, just think RAN. But don't even think about RAS now. It's not even near until week 6. So I know I said it, but they're two different things. So I feel bad that I confused you. So, but yeah, RAN is is the star of the show in the nuclear import and export cycle. So just RAND is focus on
Yeah, RAS will get their time to shine later.
week 6.
In week 6, yeah. But yeah, nuclear import is RAN is the star right now, and that's mainly what goes on with nuclear import. Nuclear export is very similar. However, its start point where we'll start is inside of the nucleus because we're exporting proteins out of the nucleus into the cytosol. So where we start here is by activating RAN with the help of GEF again. GEF will bind to RAN, cause it to become more active by switching out DP with GTP. And then now that RAN is active, it will bind to exportin. It will bind to exportin, NES, the NES signal, the nuclear export signal. Thank you, Michelle. She's pointing to the diagram for me, YouTubers. So active RAN will bind to exportin, and the cargo protein with the NES signal, nuclear export signal, will bind to exportin, which is also in that whole complex with RAN. Then RAN and that 3 protein complex will then go to the NPC, nuclear pore complex, go through it, and then GAP will allow RAN to hydrolyze its GTP, become inactive, and will let go of exportin, and then the cargo will also be released into the cytosol. And so that exportin that is now released will go back to the nuclear pore complex and back into the nucleus to continue its job of exporting proteins. That's nuclear export. Everyone good with nuclear import and export?
No question.
So now we will speed through. Oh, yes.
So that one little piece that gets left at the end is what continues to create the process.
So export and importing can do it again and again in the cycle. It has to be done many times. But remember, the whole purpose of this entire cycle is to deliver this protein outside the nucleus. So once that job is done, then the cycle starts back up. That makes sense.
Okay, so now we will go through gene expression regulation. It also is kind of like a quick section, so don't worry. I don't think we'll go over time too much, maybe just like 5 more minutes.
Then we're getting to the problem set stuff, good stuff.
Yes. So quickly, just going through the central dogma. Gene expression is how we produce proteins, and where it starts is with DNA. For eukaryotes, transcription happens in the nucleus while translation happens in the cytosol. However, for prokaryotes, there's no nucleus, so transcription and translation is done at the same time in the cytosol for prokaryotes. Eukaryotes, it happens in different places. And so that diagram to the side will go into more detail about. And there's also different controls that regulate each step of this: transcription, translation, and then also protein control as well. So just continuing on, DNA is where it starts. So DNA is read by RNA polymerase to make mRNA. And then mRNA is read by the ribosome. This is general, we'll go into more detail about this, but then the mRNA is read by the ribosome in the cytosol, and the mRNA will be translated into an amino acid sequence, and then that amino acid sequence will then fold into a fully folded protein, and how it folds together is through those R group interactions that we talked about in the first couple weeks of class. And so that's kind of just like a brief overview. After everything has been fully folded, there's also post-translational control. I won't go into a lot of detail about it because it's just listed there so you can read it. But basically, yeah, there's just like some control that you should know about that happens post-translationally. These are all additions that happen. Oh, okay, so just continuing, we're going to talk more about gene expression regulation in eukaryotes first. So transcriptional regulation. So what you should know about here are that there are different transcription factors that will regulate when and how a gene will express. So what you're looking at in this diagram is DNA, right? The green part is a ribosome. Yeah, the green part is a ribosome. But in order for that ribosome to attach and read and transcribe the gene, the DNA, transcription factors, that promoter region needs to be there. Transcription factors are just other proteins that aid um, the that aid in gene expression to occur. So those transcription factors will bind to the promoter region. Those proteins will bind to the promoter region and will aid the ribosome to read and express the gene. And basically what you need to know here are those transcription factors are also called enhancers. So when they are positively regulating, that means that the RNA polymerase is going to bind more. Oh, oops. That green thing is not a ribosome. I think I said that earlier. It's an RNA polymerase, which is reading the gene and expressing it. The green, it's not a ribosome. So those transcription factors, when it's positively regulating that RNA polymerase, that means the RNA polymerase is binding more to the gene, and that means you're getting more transcription to happen and more gene expression. However, if you have negative regulation, that means RNA polymerase will not bind as much. You won't get gene transcription and no gene expression. And that's basically transcriptional control. And so continuing, there's also.
Sorry, I realized the diagram was blocking off the word, so we're gonna fix it.
Thank you. Okay, so there's more that goes into transcriptional regulation. We went into transcription factors and those proteins that help aid in RNA polymerase binding, right? And that also changes gene expression. But there's another thing that also regulates gene expression and that controls. And that is if the DNA is in an open state versus a closed state. So chromatin, as you know, and as Laura mentioned, Professor Francis mentioned, chromatin wraps around histones and is packaged into forms, either in an open state euchromatin or heterochromatin, a closed state. And so when it's in an open state, RNA polymerase can more easily bind to DNA in order to express. But in a closed state, you can see how all the DNA is closely packed together, all the histones are closely packed together. It's harder for RNA polymerase to find a space in the DNA to bind to, and for those transcription factors to bind to the DNA in order to express genes. So those are two ways. You guys have any questions about gene expression control there? It's basically just you have an open state of chromatin and a closed state of chromatin, and then you also have those transcription factors that aid in gene expression, that positively regulate it and then negatively regulate gene expression. Okay, and then after we have passed transcription, now we have RNA modification and processing. So after you go through transcription, your product that you have is this newly made RNA. But this newly made RNA, it has introns, it has exons, but it still needs a methylguanine 5' cap to be added to that RNA. And that cap needs to be added in order to prevent nucleases from recognizing the RNA as something foreign and then cleaving it. There are nucleases that exist in the nucleus to prevent from any foreign things from existing in the nucleus in general. In prokaryotes, there are things that infect the DNA and will insert themselves into the DNA. But for eukaryotes, that will be problematic if viruses will come in, for example, our like our RNA coming in and inserting its own DNA into our DNA, kind of like COVID is an example. But that's basically something that you don't want. So but to go back to what we're talking about here, the 5' methylguanine cap prevents nucleases from recognizing the RNA and from chewing it up. What also prevents the RNA from being chewed up is a poly A tail. And this also prevents the RNA from being degraded. And that's about it. In order for the mRNA to leave the nucleus, it needs to have this 5' cap and the poly A tail. Otherwise, if it doesn't have either of these and it leaves the nucleus, then it will immediately be destroyed by nucleases or other proteins, lysosomes, for example. Yeah. Does that make sense to everybody? The reason why I went from RNA to mRNA, RNA is what we call it when it's fresh out of transcription. But mRNA is when it finally has that 5' cap and the poly A tail. Also, what's important to understand about RNA modification, before it is considered mRNA, what also happens in addition for that 5' cap adding and that poly A tail is that introns are removed from the RNA. And how that happens is through other binding proteins that will bind to the intron. I don't think we go into detail about those proteins that help with intron removal. So you guys don't have to know about that. You can just know that they're called spliceosomes. They're enzymes that will bind to introns in order to cut them out. There's also something called alternative splicing, or it's called exon skipping. Basically, spliceosomes can cut different introns in or out in order to cause different proteins to be made. And that's how you get a lot of different proteins. Like there's more diversity of proteins due to alternative splicing. Basically, like if you see in this diagram, there are 3 different exons, which the arrows are pointing to. But with alternative splicing, the mature mRNA, instead of having 3 exons, the mature mRNA will instead have like 2 exons or maybe just 1 exon, and that will cause different proteins to be made. So like from one gene, you can get like a variety of different proteins due to alternative splicing. So now going into mRNA stability, this also goes into how fast mRNA is degraded. Like all things in the cell, there is a lifespan to also mRNA. RNAi is something that is also related in regulation of mRNAs, but that's something we'll talk about like later in the course. So you don't have to worry about RNAi, but just keep it in the back of your head that like it also regulates mRNA and its presence in the cell. But yeah, if we go now, we'll go to translational control. Yeah, but basically translational control happens after the mRNA is made. There's different things that go on here. There are different enzymes that can degrade mRNA so that if there's more degradation of mRNA, for example, then you can expect that proteins won't be made from that mRNA because the mRNA is getting degraded before it can make proteins. Also, other post-translational control also includes what the activity of proteins.
And post-translational modifications from last week. An example of that. Refer to those slides.
Yeah, I don't think there's more to be added here. There are different controls that also aid in proteins getting degraded, like proteases, different enzymes that will, or different genes can upregulate whether a protein will get degraded more or less. And then same thing with mRNA. There are different genes that can be expressed that will regulate whether mRNA will be more present or get degraded more.
And even like after a protein has been created, so post-translation modifications, like tagging it with ubiquitin and sending it to the proteasome, or ubiquitin-mediated proteolysis, so destroying proteins that are tagged with ubiquitin, phosphorylation, methylation, things of that nature. Those would be considered to be post-translational modifications.
Yeah, and the last thing just to keep in mind too, the only thing that's different with prokaryotes is that because they don't have a nucleus, they really don't have transcriptional control, and RNA processing is also different. They don't need mRNA transport and localization because translation and transcription both happen in the cytosol and at the same time for prokaryotes. But everything else down the line in terms of control with like mRNA degradation, protein activation or inactivation, protein degradation, everything down the line is pretty much the same for prokaryotes. But everything above happens a little different for prokaryotes. But yeah.
Okay, so that is it for the review session. Give me 1 second.